Thank you, Nicolas, for the introduction. As he said, my name is Sandra GALEA-OUTON. I'm second-year PhD student at the LBE of INRAE, and I want to thank to the organizers of this congress to allow me to present my work titled High Biological Methane Potential of Photosynthetic Granules when Grown in Sequencing Batch Reactor. I guess that you are asking yourself what's actually a photosynthetic granule, but well, I don't want to make you a spoiler, so I will explain that later, and I will introduce you first the general frame, which is the phototrophic systems. So the phototrophic systems have higher biomass yields than strictly heterotrophic systems like activity sludge, which is a typical biomass that you can find in the wastewater treatment plants. This happens because the CO2 This happens because the CO2 from the organic carbon catabolites is also incorporated in the freshly grown biomass, actually the photosynthetic biomass. So this produces an excess of biomass that can be an advantage if we identify uses for it. So now it comes to the photosynthetic granules. In this study, we call them oxygenic photogranules, or OPGs. And these are granular biofilms that are made of phototrophic microorganisms, especially cyanobacteria, and heterotrophic microorganisms that interact syntrophically. So the phototrophs consume light and CO2, and they produce oxygen and organic matter like EPS. These are consumed by the heterotrophs that after produce CO2, which is again assimilated by the heterotrophs. In these pictures, you can see a diversity of sizes, and morphologies of different OPGs under stereo microscope. This biomass is especially interesting for several processes like wastewater treatment, because they have some interesting characteristics like good settability that make it easier for biomass recovery. Also, the biomass can be reused. And as they produce oxygen, there is no need of external aeration, and that means a lower energy requirement in the aerobic processes. So what happens if we use the OPGs in processes like, for example, the wastewater treatment? We will get an excess of biomass. What we propose is to use this excess of biomass as a substrate to produce methane through anaerobic digestion, of course. So, in order to obtain the maximum advantage of this granular biomass, we wondered what conditions, what culture conditions in a sequencing batch reactor would be the best. So, our objective is to identify those reactor parameters that may influence more in the BMP of the granules. This is the biochemical methane potential, which is an indicator of the biodegradability. That means that when the more biodegradable is the biomass, the more methane we can obtain from the fermentation of this biomass. So how to identify the relationship between the BMP and the reactor parameters? Well, we grow the OPGs under different conditions in the reactors to harvest biomass that was freeze-dried and after analyzed with near-infrared spectroscopy on air. Then we use this spectra to predict the methane potential using a model from Leicester that was already proved. In addition, the NIR also allows us to obtain a biochemical characterization of this biomass. And after we analyze this data, the BMP data and the parameters, through a PLS analysis, a partial S-square regression. This allows us to extract the parameters that have more influence on the BMP. So we had 2 reactors that were working for 400 days, and with some constant parameters, which were the media, that was a sterile synthetic wastewater with acetate current source, the stirring, and the working and removal volumes. The cycle length was changing, so the hydraulic retention time was depending only on the cycle length, but not in the volumes. On the other side, we had all the 6 reactor parameters that were changing, and these parameters are the ones that we study with the partial least square. So these are the light intensity, the light regime that changed from the constant light to up to 1 hour of darkness per cycle. Cycle length, as I said before. The initial concentration of chemical oxygen demand, which is due to the acetate present in the media. And this is also the maximum concentration of CO2 that can be reachable in every cycle. The load, that depends on the cycle length and also on the concentration. And finally, the biomass concentration that was analyzed through total suspended solids. So along all that time, I harvested 61 samples to analyze with the NIR and predict the methane. I want to remark that these samples were always took when the organic carbon in the bulk phase of the reactors were 0. So we can ensure that the BMP value is due to only the biomass and not remaining acetate in the media. So these are the first results. Here you can see the average BMP of activity sludge that represents the heterotrophic population that we can find inside of the granules. The average BMP of cyanobacteria cyanobacteria cultures that were initially isolated from granules. And on the other hand, the average BMP of granules that were grown in the reactors. So you can see these are higher, and this can be due to higher lipid proportion that we observe also by NIR. This might be related to the specific growth conditions in the reactor and not simply to the presence of these relative biomasses. Here I also want to remark, as I say, this is the average BMP of the granules of those 400 days of the reactors. So here we cannot see the effect of every parameter, but here we can. In every parameter, every combination of parameter keep a condition in the reactor for at least 3 days, as the reactor can change quite fast. So in the partial least square regression, the PLS, we can observe that only the load and the maximum CO2, that by the way I will keep calling them just organic load or organic carbon, because they are quite related in a way. So these 2 parameters are the only ones that have a positive influence on the BMP, which is displayed in this red arrow for an easier understanding. Both principal components explain almost 40% of the variance on the BMP. So these results suggest that the granules are able to convert the acetate into starch compounds, which makes sense with the higher lipid proportion that we found. However, we miss some parameters that may explain a higher variance on the BMP. This could be, for example, the biochemical composition that we haven't included in the PLS. So to test this idea, the effect of the organic load, we made this experiment. We exposed the reactors to 3 days of starvation, and we could see a clear decrease on the BMP value. So this suggests that in these 3 days, the storage compounds were consumed, and after 10 to 20 days, they were recovered. So finally, the conclusions. The oxygenic photogranules grown in sequencing batch reactor have, on average, higher biochemical methane potential than activity sludge or cyanobacteria, and this is due to the growth conditions. The storage compounds can be consumed in 3 starvation days, leading to lower BMP that after can be recovered. Among the SBR parameters that were tested in this study, just the organic carbon load has the highest influence on the BMP. But however, the PLS model has a low percentage of variance on the BMP data. So we think on ways to improve this model, and we suggest, of course, more data, but also maybe add some other parameters like the macromolecular composition or the microbial composition, like the proportion of heterotrophs or phototrophs. So that was all. Thank you very much. If you have any question.